Everyone, so we decided to do our project on the effectiveness of added nitrogen and bacteria on the rate of bioremediation in oil spills, since this is still an active environmental problem present in our society. It is estimated that about 8.4 million tons of petroleum waste pollutes the ocean yearly. Nonetheless, several genera of microorganisms have been identified with being able to convert polycyclic aromatic hydrocarbons, or PAHs, among other toxic carbon substances found in petroleum products, into biomass, carbon dioxide, and water, a process known as bioremediation. In the Pseudomonas genera specifically, the operons NaH, PAH, DOX, PHN, PhD, and NAG have been identified with PAH degradation. Though much is still to be discovered on the process, bioremediation efforts have already been applied in historical petroleum disasters, such as the Exxon Valdez spill of 1989 and the more recent BP Deepwater Horizon disaster of 2010. Biostimulation has oftentimes been coupled to enhance bioremediation progress. This typically involves adding fertilizer to the contamination sites in order to increase critical nutrient levels of phosphorus, nitrogen, and/or carbon for the hydrocarbon degraders. Of course, we understand that pH, oxygen, moisture content, temperature, metal ions, and soil type are additional important factors in affecting this process.
In this experiment, we will try to determine how the addition of different factors, nitrogen and Pseudomonas aeruginosa, affect the rate of oil bioremediation. Based on research, we hypothesize that adding both nitrogen and bacteria will increase the rate of bioremediation compared to the factors individually. We chose to conduct the study on a shoreline instead of on open waters, as many laboratory experiments and field trials have been conducted that conclude that bioremediation can be useful on shorelines. Amongst the studied factors that affect the rate of bioremediation, we chose studied nitrogen concentration since degradation is proportional to the average amount of nitrogen per unit of oil. Additionally, both nitrogen and phosphorus concentrations have been highlighted in numerous studies as crucial limiting factors impacting the effectiveness of bioremediation. According to one study, the minimum nitrate concentration needed to support growth of alkane degraders ranges between 0.5 and 2.5 milligrams of nitrogen per liter. Therefore, we will be using 1.5 milligrams during a hypothetical experiment. Also, since the same study uses sodium nitrate as their nitrogen source, we will be using it as well. Pseudomonas aeruginosa was chosen as our microbe since its PAH's degrading ability has been highlighted as being the most thoroughly studied thus far.
To perform this experiment, we set up 10 buckets, each full of 1 liter of seawater from Clearwater Beach, and any debris in the water was filtered out. There are 2 buckets for each of the 5 treatments. One contains 1 liter of just seawater, serving as our negative control. Another contains 0.2 liters of oil, serving as our positive control. Another contains 0.2 liters of oil and 1.5 milligrams of nitrogen. Another contains 0.2 liters of oil and is inoculated with a strain of Pseudomonas. And the last contains 0.2 liters of oil, was inoculated with the bacteria, and also contains the added nitrogen. Once a week, each of the 10 buckets were replenished, and every 10 days for 100 days, a 1 microliter water sample was extracted from each bucket and tested using gas chromatography. Looking at the graphs, we can see clear results. The graph on the left are the chromatographs from the water samples after 20 days. The lowest graph, which is the bucket treated with oil, Pseudomonas, and the nitrogen, has the greatest decrease in frequency and magnitude of spikes, compared with the other treatment buckets, which still show a large amount of chemical presence in the oil left over in the buckets. The graph on the right shows the amount of alkanes and aromatics in the samples over the 100 days. This graph shows the rate of biodegradation of all the treatments, and it can be clearly seen that treatments of both bacteria and nutrients, the square and circle lines, removed more, in some cases all of the alkanes and aromatics over the 100 days faster than the control. This shows that treatments of bacteria and nutrients can biodegrade at a faster rate than the natural rate of the environment.
Based on the literature we found and the results of the experiment, it is evident that the combination of the Pseudomonas aeruginosa bacteria and the addition of nitrogen induces the highest rate of biodegradation. The data demonstrates statistically significant bioremediation rate enhancement when the natural nutrient levels are increased using fertilizers. The added nutrients within these fertilizers stimulated the rapid multiplication of microbes, which simultaneously increased cleanup rates. It's important to note that the acceleration of biodegradation by the addition of fertilizers to the polluted area was effective only after the level of contamination had been reduced, either by natural elimination or by mechanical recovery. Bioremediation is a cost-effective and environmentally friendly tool. However, it should not be considered a miracle cure, but rather a complementary technique to more conventional methods of treating pollution. To combat this, scientists are currently turning towards engineered microbes. These genetically engineered microbes have the potential for bioremediation applications, considering that the metabolic pathways which limit naturally growing bacteria can be modified, using bacteria that can bring about optimal and enhanced degradation using fewer resources. Given this information, further research should be conducted to measure the effectiveness of engineered microbes and the enhancement of bioremediation, especially in locations where nutrients are scarce, such as low and mid-latitude ocean waters.